This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome to an exploration of one of the most diverse and fascinating groups of birds on our planet, the passerines, or perching birds. From the familiar chirp of a sparrow to the vibrant colors of a tanager, passerines are a constant presence in our world, their songs filling our gardens and forests. But have you ever stopped to wonder where these remarkable birds came from? How did they evolve their incredible diversity of forms, behaviors, and songs? This journey of discovery will delve into the deep history of passerines, unraveling their evolutionary story in easy-to-understand language. Over the course of this exploration, we'll travel back in time, tracing the passerine lineage from its ancient origins on the supercontinent Gondwana to its current global dominance. We'll uncover the geological forces that shaped their evolution, the genetic clues that reveal their family relationships, and the remarkable adaptations that have allowed them to thrive in virtually every corner of the Earth. Prepare to be amazed by the story of the perching foot, a marvel of biomechanics that allows passerines to effortlessly cling to branches. We'll explore the diverse nesting strategies that have enabled them to occupy a wide range of habitats, from simple cavities in the ground to elaborate hanging structures woven from grasses and twigs. We'll delve into the secrets of the syrinx, the unique vocal organ that allows songbirds to produce their complex melodies and mimic a vast array of sounds. This journey will take us from the fossil beds of Australia and North America, where paleontologists unearthed the delicate bones of ancient passerines, to the laboratories of geneticists, who unravel the secrets hidden within their DNA. We'll explore the biogeographic puzzles that challenge our understanding of their dispersal across continents, and we'll examine the ecological forces that have shaped their remarkable diversification. By the end of this exploration, you'll have a deeper appreciation for the intricate evolutionary history of passerines, from their ancient origins to their current global dominance. You'll understand the key adaptations that have allowed them to thrive in such a wide range of environments, and you'll gain insights into the ongoing research that continues to rewrite their evolutionary story. So, join us as we embark on this fascinating journey into the world of passerines, a symphony of feathers played out over millions of years. Chapter 1. From Gondwana to the Globe, tracing the passerine's first flight. Imagine our world not as it is today, but as a single, massive landmass, a supercontinent called Gondwana. This colossal expanse of land, incorporating what we now know as South America, Africa, India, Australia, Antarctica, and New Zealand, was a world teeming with life unlike anything we see today. Giant ferns and conifers dominated the landscape, while early mammals and reptiles, often much larger than their modern counterparts, roamed the earth. This was the setting, millions of years ago, where the remarkable story of passerines, or perching birds, began. Gondwana was a land of extremes. Vast deserts stretched across its interior, while lush rainforests flourished along its coastlines. Volcanoes erupted, shaping the land and influencing the climate. This dynamic environment played a crucial role in the evolution of life on Earth, including the ancestors of our modern birds. As Gondwana slowly began to break apart, driven by the powerful forces of plate tectonics, its inhabitants were faced with new challenges and opportunities. The shifting landmasses created barriers, isolating populations and fostering the evolution of unique characteristics. One such event, around 82 million years ago, was the geological separation of New Zealand from the remnants of Gondwana. This isolation created a unique evolutionary laboratory in the South Pacific. Imagine this newly isolated island, adrift in the vast ocean, a haven for delicate creatures like the early passerines. Without the constant threat of large land mammals and reptiles that dominated other parts of Gondwana, these small birds could thrive. Their smaller size, once a vulnerability, became an advantage, allowing them to exploit resources unavailable to larger animals. With fewer predators, they could diversify and evolve new forms, setting the stage for their future global dominance. Fast forward to around 47 million years ago, and we find the first true passerines emerging in the South Pacific, in the vicinity of what is now Australia and New Zealand. These early perching birds, though small, were incredibly diverse. Their evolutionary success can be attributed to a unique combination of characteristics: their small size, their ability to perch, and their knack for building nests in a variety of locations. Today, passerines are a testament to this early diversification. They represent over 60% of all bird species and occupy a stunning array of ecological niches. From the cheerful melodies of canaries to the ubiquitous chirping of house sparrows, passerines are a constant presence in our world. Even the vibrant green-headed tanager, a jewel of the Brazilian Atlantic forest with its iridescent green head and turquoise wings, belongs to this remarkable group. Among this diverse order, the oldest family is believed to be the Acanthisididae, also known as New Zealand wrens. These tiny birds, found only in the mountainous regions of New Zealand, offer a glimpse into the ancient past of passerines. Their small size, simple songs, and unique anatomical features distinguish them from other perching birds, making them a crucial piece in the puzzle of passerine evolution. Their continued existence in New Zealand, isolated from the rest of the world, reinforces the importance of geographic isolation in shaping the evolution of this diverse group. The early passerines eventually gave rise to two other ancient suborders, Turani and Passerine. Turani, meaning tyrant birds, includes species like the great kiskadee, known for its loud, distinctive call, and the neotropical bellbird, with its remarkable resonating song. These birds are primarily found in South America, although some species have ventured into North America. Passerine, on the other hand, meaning sparrow birds, is a larger group with a wider global distribution. This suborder includes familiar birds like thrushes, known for their beautiful songs, and siskins, small finches with distinctive yellow markings. From these three ancestral groups, the New Zealand wrens, the Turani, and the Passerine, descended the more than 6,000 passerine species that grace our planet today. The evolutionary journey of passerines, however, stretches back even further. The very first birds, arising over 150 million years ago, were themselves descendants of feathered dinosaurs. This incredible transformation, from terrestrial reptiles to feathered flyers, is a testament to the power of evolution. While much of this early evolutionary process remains shrouded in mystery, we know that it involved a series of remarkable changes, including the development of feathers, wings, and the ability to fly. The story of how these feathered dinosaurs evolved into the graceful birds we see today is a subject of ongoing research and discovery, and one we will explore further in the chapters to come. Chapter 2. Feathered Footprints in Time. Fossil Clues to Passerine Ancestry. The delicate bones of passerines, so perfectly adapted for perching and flight, pose a significant challenge for paleontologists. These fragile structures rarely withstand the test of time, making passerine fossils a rare and precious find. Yet, despite their scarcity, these fossilized remnants offer invaluable glimpses into the deep history of this diverse bird group, illuminating their origins, adaptations, and intricate journey across the globe. The oldest undisputed passerine fossils date back to the Eocene epoch, approximately 50 million years ago. Discoveries in Mergen, Australia, have yielded fragmented remains, hinting at early passerine forms. Half a world away, in the Green River Formation of Wyoming, USA, another early Eocene site has revealed remarkably preserved, near-complete skeletons of early passerines. One such find is Eozygodactylus americanus, a small bird whose skeletal structure provides clues to the evolution of perching adaptations. The unique morphology of its feet, with a specialized arrangement of toes and the development of a strong hallux, or hind toe, suggests an increasing ability to grasp branches, a crucial step towards the refined perching abilities seen in modern passerines. From the same North American Eocene deposits comes a truly remarkable fossil, Phoropinarium. This near-complete specimen, also dating back to the early Eocene, exhibits a fascinating mosaic of features. While some characteristics align with general early passerine morphology, others show an unexpected connection to modern turacos, a family of brightly colored, fruit-eating birds now found exclusively in sub-Saharan Africa. The presence of an unfused furcula, or wishbone, a short, stout bill with a broad nasal process, and other skeletal similarities point towards this surprising transcontinental link. The discovery of Foropinarium not only challenges our understanding of early passerine biogeography but also provides the first well-supported evidence of a stem turrico, offering a crucial calibration point for molecular dating studies aimed at unraveling the timing of bird evolution. The Eocene epoch also holds other important clues to early passerine diversification. In Europe, fossils like Wieslokia, Jamna, Rhizoviornis, and Krosnornis, discovered in various locations including Germany and Poland, paint a picture of a diverse early Eocene avian fauna. These fossils represent both modern and extinct lineages, with some showing affinities to Sabosons, a group of passerines with a simpler vocal structure than their songbird relatives. The presence of these early passerine relatives in Europe, far from their modern tropical distribution, suggests a more complex biogeographic history than previously imagined. Some fossils, like a humerus fragment resembling a broadbull, a group now found in the Old World tropics, found in early Miocene deposits in Germany, further support the idea of early widespread distribution for lineages now restricted to specific regions. Moving into the Miocene epoch, younger fossil sites continue to provide pieces of the passerine puzzle. The Bathans Formation in Otago, New Zealand, has yielded Miocene fossils including leg bones from birds of varying sizes, some comparable to a modern tui, others closer to a saddleback. These fossils contribute to our understanding of passerine evolution within the isolated New Zealand avifauna, demonstrating the persistence and diversification of passerines in this unique island environment. While fossils like Foropinarium provide key insights, the fossil record for passerines, especially early forms, is notoriously incomplete. Their small size and the delicate nature of their bones make fossilization a rare event. The majority of passerine fossils are fragmentary, often consisting of isolated bones rather than complete skeletons. This makes interpretation challenging, as researchers must piece together the evolutionary story from limited evidence. Furthermore, the relatively recent emergence of passerines, compared to other bird groups, means that the window of time for potential fossilization is shorter. The preservation of fossils also depends on specific environmental conditions, such as rapid burial and the presence of fine-grained sediments, which are not always present in habitats favored by passerines. Despite these challenges, fossil evidence remains a cornerstone of our understanding of passerine evolution. Fossils provide tangible evidence of past life, allowing researchers to directly observe the morphology of extinct species. They offer crucial calibration points for molecular clocks, helping to refine the timeline of passerine diversification. By comparing the anatomical features of fossil passerines to those of modern birds, scientists can infer evolutionary relationships and trace the development of key adaptations like perching, flight, and bill shape. Moreover, the geographic distribution of fossils offers valuable clues to the biogeographic history of passerines. Unexpected fossil distributions, like the turrico-like Foropinarium in North America, challenge traditional narratives and suggest a more dynamic and complex history of dispersal and extinction than previously imagined. As new fossil discoveries continue to emerge, they will undoubtedly further refine our understanding of the passerine family tree and their remarkable journey from Gondwana to their current global dominance. Chapter 3. The DNA Detectives: Rewriting the Passerine Family History. The family history of passerines, the largest order of birds, was, until recently, a blurry photograph of the past. Scientists, like dedicated genealogists, relied on observable characteristics, the shape of a beak, the color of plumage, the structure of a foot, to piece together the relationships between different species. But these physical traits, molded by similar lifestyles and environments, often painted a misleading picture of evolutionary kinship. Then came the DNA revolution, transforming our blurry snapshot into a high-definition portrait of passerine evolution. Imagine holding a tiny feather in your hand, perhaps a vibrant green one shed by a green-headed tanager flitting through the Brazilian Atlantic forest, or a sleek black one dropped by a tui, a bird with a haunting song unique to New Zealand. Within these seemingly insignificant fragments lies a treasure trove of information, DNA, the very blueprint of life. By analyzing the genetic material within these feathers, and comparing it to DNA from hundreds of other bird specimens from around the world, scientists have gained unprecedented insights into the passerine family tree. One of the most powerful tools in this DNA detective work is the analysis of ultra-conserved genomic loci. These are stretches of DNA that have remained remarkably similar across vast evolutionary timescales, acting as anchors in the swirling sea of genetic change. By comparing these loci across different passerine families, researchers can pinpoint the branching points in their evolutionary history, revealing who is most closely related to whom. This technique has helped resolve some of the most challenging branches of the passerine family tree, entangling relationships that were previously obscured by convergent evolution. Another key piece of the genetic puzzle comes from mitochondrial genomes. These small, circular pieces of DNA, residing within the mitochondria, the powerhouses of the cell, are inherited solely from the mother. Mitochondrial DNA evolves at a different rate than nuclear DNA, providing an additional layer of information for reconstructing evolutionary relationships. Analyzing mitochondrial genomes has been particularly useful in resolving relationships at the species level and has even shed light on the timing of major evolutionary events. These genetic investigations have led to some surprising discoveries, overturning long-held assumptions about passerine evolution. For instance, the discovery of a new family of passerines, the Hyliidae, in Africa, was a complete surprise. These birds, previously classified within other families, were found to possess unique genetic markers, distinguishing them as a distinct evolutionary lineage. Similarly, the taxonomic status of several other families, previously debated, has been confirmed through DNA analysis, solidifying our understanding of their place in the passerine family tree. Perhaps the most dramatic rewrite of passerine history involves the timing of their diversification. For years, the prevailing estimate placed the origin of passerines around 82 million years ago, coinciding with the separation of New Zealand from Gondwana. The narrative was that these small birds thrived in New Zealand's isolated environment, free from the pressures of mammalian predators. However, this timeline clashed with the fossil record, with the oldest passerine fossils being significantly younger. The genomic data, combined with biogeographic information and careful fossil calibration, tells a different story. By incorporating DNA from a large and diverse sample of bird specimens, including species like the green-headed tanager and the tui, and calibrating their molecular clocks with well-identified passerine fossils like Eozygodactylus americanus, researchers have pushed back the passerine origin to around 47 million years ago, a staggering 35 million years younger than the previous estimate. This revised timeline paints a more nuanced picture of passerine evolution, suggesting that their diversification was not a simple consequence of Gondwanan vicariance but a more complex process involving dispersal, radiation, and adaptation to changing environments. These genomic revelations have far-reaching implications for understanding bird evolution as a whole. The placement of the New Zealand wrens as the sister taxon to all other passerines strengthens the idea of a southern hemisphere origin for this group. The discovery of Phoropinarium, a stem turrico in the Eocene of North America, challenges the traditional narrative of a strictly Gondwanan diversification for many bird lineages. These findings, combined with similar biogeographic puzzles presented by other bird groups, highlight the importance of integrating fossil data, genomic analysis, and biogeographic information to paint a complete picture of the avian family tree. The story of passerine evolution is far from over. Projects like Open Wings, an international collaboration of ornithologists, are using genomic data to further refine bird relationships at the species and genus level. By combining data from around the world, Open Wings aims to create an even more detailed and accurate portrait of the passerine family, revealing the intricate details of their evolutionary journey and their remarkable success in colonizing virtually every corner of our planet. Chapter 4. A Nest for Every Need: How Homes Shaped the Passerine Story. Nesting behavior, more than just a biological necessity for raising young, has profoundly shaped the evolutionary trajectory of passerines, the most diverse order of birds. Their remarkable diversification, encompassing over 60% of all bird species, is intricately linked to the evolution of diverse nesting strategies, from simple cavities scratched into the earth to elaborate hanging structures and architectural marvels woven from grasses and twigs. This flexibility in nest design and placement has allowed passerines to exploit an unparalleled range of habitats, contributing to their remarkable success. The evolutionary history of passerine nests traces back to three basic nest types: cavity nests, open nests, and domed nests. Evidence suggests that even the earliest passerines, those first emerging in the South Pacific near Australia and New Zealand, likely constructed all three of these basic nest forms. This early diversity in nesting strategies provided a foundation for their subsequent radiation into new environments and ecological niches. Cavity nests, whether tucked away in tree hollows, burrows in the ground, or crevices in rock faces, offer a secure refuge for eggs and nestlings, protecting them from predators and the elements. This nest type has been particularly successful in higher latitudes, where whole nesting species like tits, parody, benefit from the insulation provided by their enclosed nests. This insulation allows them to lay larger clutches than open nesting species, increasing their reproductive potential in challenging environments. The green-headed tanager, Tangara celadon, a vibrant resident of the Brazilian Atlantic forest, exemplifies another advantage of cavity nests: camouflage. Their nests, hidden within tree hollows or dense foliage, blend seamlessly with their surroundings, providing an additional layer of protection from predators. Open nests, in contrast, are constructed on branches, in forks of trees, or on other exposed surfaces. These nests, ranging from simple cups to substantial platforms, often incorporate a variety of materials, including twigs, leaves, grasses, and even mud. While open nests may be more vulnerable to predation and weather, they offer advantages in terms of access to food resources and ventilation. The diversity of open nest designs reflects the varied ecological roles of their builders. For instance, some foliage-gleaning furnarids, a family of ovenbirds found in dense forests, construct relatively simple open cups nestled among leaves, providing easy access to their insect prey. Domed nests, a unique feature more prevalent among passerines than other bird groups, represent an intriguing evolutionary innovation. These nests, with their constructed roofs, offer enhanced protection from predators and weather, particularly beneficial for the smallest passerine species. The ovenbirds, Furnariidae, a large and diverse family found primarily in South America, showcase the remarkable evolutionary plasticity of domed nests. The rufous hornero, Furnarius rufus, a common sight in open grasslands, constructs a sturdy, oven-like nest from mud and clay. This robust structure, placed on exposed branches or fence posts, provides excellent insulation and protection from predators, allowing the hornero to successfully raise its young in a habitat with limited natural nest sites. Contrast this with the simpler nests of some foliage-gleaning furnarids, who build less elaborate domed structures among the leaves of their forest homes, where camouflage and access to insects are more critical than insulation. Beyond these three basic forms, specialized nest types have evolved in various passerine lineages, often reflecting unique ecological challenges and opportunities. The Oropendulas, Icteridae, found in the Neotropics, construct remarkable pendulous nests woven from grasses and vines. These hanging structures, suspended from the tips of branches, offer protection from climbing predators like snakes and monkeys, allowing the Oropendulas to safely raise their young in forest canopies. Weaver birds, Placeidae, primarily found in Africa and Asia, are renowned for their intricate woven nests, often constructed in colonies. These complex structures, woven from grasses, strips of leaves, and other plant fibers, provide a secure and camouflaged haven for eggs and nestlings. The colonial nesting behavior of some weaver species, such as the southern masked weaver, Placeus vellatus, amplifies the protective benefits of their nests. The sheer number of nests in a colony can overwhelm predators, increasing the survival rate of individual nestlings. This strategy is particularly effective in open savanna habitats, where alternative nest sites are scarce and predator pressure is high. Nest site selection, just as important as nest structure, is another key factor in passerine reproductive success. Factors like camouflage, insulation, proximity to food sources, and the availability of suitable nesting materials all play a role in a bird's choice of where to build its home. For instance, lesser kestrels, Falco naumani, small migratory falcons, gather in large pre-migratory roosts in specific areas of the Balkans before their long journey to Africa. Genetic analysis of feathers shed at these roosts has revealed that birds from multiple breeding populations, including those in Italy and Greece, converge on these sites, some traveling hundreds of kilometers. These pre-migratory sites provide abundant insect food resources, allowing the kestrels to build up essential fat reserves for their migration and to undergo molt, replacing worn feathers. The diversity of nesting behaviors exhibited by passerines is a testament to their evolutionary adaptability and a key factor in their extraordinary success. Their ability to build a nest for every need, exploiting a wide range of habitats and ecological niches, has allowed them to become the most diverse group of birds on our planet. From the simple cavities of ground nesters to the intricate woven colonies of weaver birds, each nest tells a story of adaptation, survival, and the remarkable interplay between a bird and its environment. Chapter 5. The Songbird's Secret: Tuning into the Evolution of Vocal Mastery. Passerines, representing over half of the world's bird species, have not only conquered diverse habitats with their adaptable nesting strategies but also mastered the art of vocal communication. The evolution of vocal complexity, particularly in the OS signs or songbirds, is a defining characteristic of this remarkable group. Their diverse vocalizations, from simple calls to elaborate songs, play a crucial role in various aspects of their lives, including mate attraction, territorial defense, species recognition, foraging, predator avoidance, and the maintenance of social bonds. At the heart of this vocal mastery lies the syrinx, a unique vocal organ found only in birds. Located at the base of the trachea, where it splits into the two bronchi leading to the lungs, the syrinx is a complex structure composed of membranes, muscles, and cartilaginous rings. Unlike the mammalian larynx, which produces sound through the vibration of vocal cords, the syrinx generates sound through the vibration of the tympaniform membranes within the bronchial walls. Remarkably, birds can control the two sides of their syrinx independently, allowing them to produce two distinct sounds simultaneously or to rapidly switch between different notes and frequencies. This intricate control enables the production of the diverse and complex vocalizations that characterize passerines. The range of sounds passerines produce is truly astonishing. From the simple chirps and whistles of sparrows to the complex melodies of thrushes, the syrinx allows for an extraordinary range of vocal expression. The green-headed tanager, Tangara celadon, of the Brazilian Atlantic forest uses its syrinx to produce a variety of calls, including contact calls to maintain flock cohesion, alarm calls to warn of predators, and territorial songs to defend its breeding territory. The tui, Prosthemodera novaezelandiae, endemic to New Zealand, is renowned for its rich and varied song, incorporating clicks, whistles, and bell-like tones. Its syrinx allows it to produce these diverse sounds with remarkable clarity and volume, making its song one of the most distinctive in the New Zealand avifauna. The evolution of song complexity in some passerine lineages has been intricately linked to sexual selection. In many species, females prefer males with more elaborate and complex songs, driving the evolution of increasingly sophisticated vocalizations. This preference for complex songs may indicate a male's genetic quality, health, or ability to defend a territory, thus increasing the reproductive success of females who choose such males. This process of sexual selection has resulted in the beautiful and intricate songs that fill our forests and gardens, from the melodious warbling of nightingales to the elaborate mimicry of lyrebirds. The superb lyrebird, Monora novaezelandiae, of Australia is a prime example of vocal mimicry taken to an extreme. Its syrinx, combined with its remarkable vocal learning abilities, allows it to imitate an astounding variety of sounds, including the calls of other birds, the sounds of human-made objects like chainsaws and camera shutters, and even the melodies of musical instruments. This extraordinary mimicry, honed by sexual selection, serves as a display of the male lyrebird's vocal prowess and cognitive abilities, attracting females and increasing its chances of reproductive success. Vocal learning, the ability to acquire and modify vocalizations through imitation, is another remarkable characteristic of OS signs. This ability, rare in the animal kingdom, has been linked to increased brain size and development in passerines. The brains of songbirds contain specialized regions dedicated to vocal learning and production, analogous to the language centers in the human brain. These regions are larger and more developed in species with more complex songs, further supporting the link between vocal learning and brain evolution. The connection between vocal learning and brain development is evident in the complex vocal dialects found in some songbird populations. For example, white-crowned sparrows, Zonotrichia leucophrys, exhibit distinct regional dialects, with birds from different areas singing subtly different versions of the same song. These dialects are learned during a critical period in the bird's development, when young birds imitate the songs of adult males in their local population. This cultural transmission of song, facilitated by vocal learning and neural plasticity, adds another layer of complexity to passerine communication. Interestingly, vocal complexity is not limited to songs used in mate attraction and territorial defense. Passerines employ a wide range of vocal signals for various purposes. Alarm calls, often sharp and high-pitched, alert flock members to the presence of predators. Flocking calls, typically soft chirps and whistles, help maintain group cohesion during flight or foraging. Begging calls, given by nestlings, elicit feeding behavior from their parents. The green-headed tanager, for instance, uses distinct calls to communicate about different types of threats, allowing flock members to respond appropriately to specific predators. The transition from vocal communication to taste reception may seem abrupt, but the ability to detect specific tastes, like the sweetness of nectar or fruit, has also played a significant role in avian evolution, particularly in groups with specialized diets. While the common ancestor of all birds lost the ability to detect sweet tastes, some lineages have regained this ability through unique evolutionary pathways. Hummingbirds, Trochilidae, a group of nectar-feeding birds found in the Americas, offer a fascinating example of how taste receptors can evolve. While most birds lack the typical sweetness receptors found in mammals, hummingbirds have co-opted a gene originally responsible for detecting umami, or savory tastes, and transformed it into a sweetness receptor. This evolutionary repurposing of a gene, through a series of mutations, allowed hummingbirds to detect the sugars in nectar, crucial for their high-energy lifestyle. The specific genes involved in this transformation, including TUSH1R1 and TUSH1R2, have been identified through genomic analysis, revealing the molecular basis of this remarkable evolutionary adaptation. Remarkably, songbirds, like hummingbirds, have also regained the ability to detect sweet tastes. Recent research indicates that this ability arose early in songbird evolution, possibly in Australia. This discovery supports the growing body of evidence pointing towards an Australian origin for many key passerine characteristics, from vocal learning to taste reception. Unlike hummingbirds, however, songbirds have evolved a different mechanism for detecting sweetness, demonstrating the independent evolution of this ability in the two groups. This convergent evolution highlights the importance of taste detection in exploiting nectar and fruit resources, providing a selective advantage for birds with this ability. Several Australian songbirds exemplify the importance of sweet taste detection in their ecological roles. The New Holland honeyeater, Philodonyrus novaehollandiae, with its specialized brush-tipped tongue, feeds on nectar from a variety of flowers, including banksias and grevilleas. Its ability to detect sweetness allows it to efficiently locate and exploit these nectar sources, playing a crucial role in pollination. Similarly, many parrot species, such as the rainbow lorikeet, Trichoglossus mollucanus, also found in Australia, have a preference for nectar and fruits. Their ability to detect sweetness, though evolved through a different pathway than songbirds or hummingbirds, allows them to thrive on these sugary food sources. The evolution of sweet taste detection in these Australian birds underscores the interplay between sensory perception, diet, and ecological adaptation, highlighting the diverse ways that passerines have tuned into their environment to achieve their remarkable evolutionary success. Chapter 6. A Symphony of Feathers, Unraveling the Passerine Enigma. Passerines, the perching birds, are a global phenomenon. Their melodies fill our gardens, their vibrant colors flash through forests, and their bustling activity enlivens even the most mundane landscapes. From the tiny short-tailed pygmy tyrant, weighing a mere 4.2 grams, to the hefty thick-billed raven, tipping the scales at over 1.5 kilograms, passerines showcase an astounding diversity in size, form, and lifestyle. Their evolutionary success story, a symphony of feathers played out over millions of years, is a testament to the power of adaptation and the intricate interplay between geology, genetics, and ecological opportunity. This remarkable group, comprising over 6,000 species, represents over 60% of all bird species on Earth. They have conquered virtually every terrestrial habitat, from the lush rainforests of the Amazon to the arid deserts of Australia, from the high mountains of the Himalayas to the bustling cities of Europe and North America. Only Antarctica remains untouched by their vibrant presence. This global dominance is a testament to their evolutionary flexibility, honed over millions of years through a combination of key adaptations. One of the most significant factors in passerine success is their small size. This characteristic, often a vulnerability in the animal kingdom, has become a key to their diversification. Their diminutive stature allows them to exploit a vast array of ecological niches, from gleaning insects from leaves to sipping nectar from flowers, from cracking seeds to catching flying insects on the wing. The green-headed tanager, Tangara celadon, a vibrant jewel of the Brazilian Atlantic forest, exemplifies this adaptability. Its small size allows it to navigate the dense foliage, gleaning insects and fruits from the forest understory. Contrast this with the tui, Prosthemodera novaezelandiae, of New Zealand, a larger passerine that uses its robust build and powerful beak to access nectar from deep within flowers, playing a crucial role in pollination. Passerines are also masters of perching, a skill enabled by the unique anatomy of their feet. 3 toes point forward, while a long, opposable hind toe, called the hallux, allows them to securely grip branches of varying sizes. This specialized foot structure, combined with the flexibility afforded by their unfused leg tendons, allows passerines to easily maintain an upright posture on branches, even while sleeping. For tree-climbing species, like tree creepers and wood creepers, stiff tail feathers provide additional support, acting as props against the trunk. This adaptation, coupled with their strong claws and flexible toes, allows them to effortlessly ascend vertical surfaces in search of insects and other food. The evolutionary flexibility of the passerine foot is further demonstrated by the diverse functions of the tail. While most passerines have 12 tail feathers, some species, like the superb lyrebird, possess 16, while others have fewer. In some species, like the long-tailed widow bird, the tail has been elaborated into extravagant ornaments, used by males to attract females. Nesting behavior is another cornerstone of passerine success. Their ability to construct diverse nests in a variety of locations has allowed them to occupy a wide range of habitats, from the ground to the highest treetops. Cavity nests, like those of the green-headed tanager, provide a safe haven for eggs and young, protecting them from predators and harsh weather. Open cup nests, common among species like the American robin, offer easy access to food resources and good ventilation. Domed nests, a characteristic more common among passerines than other bird groups, provide additional protection from predators and the elements, particularly beneficial for the smallest species. The ovenbirds, Furnariidae, of South America exemplify the diversity of domed nests, with some species building elaborate oven-like structures from mud, while others construct simpler domed nests hidden among foliage. Passerine reproduction is further characterized by the laying of colored eggs, a trait thought to aid in camouflage or species recognition. Clutch sizes vary widely, reflecting the diverse life histories of different species. Some larger Australian passerines, like lyrebirds and scrub robins, lay only a single egg, investing heavily in parental care. In contrast, smaller passerines in warmer climates may lay clutches of 2 to 5 eggs, while whole nesting species in higher latitudes can lay up to a dozen. This variation in clutch size reflects a complex interplay of factors, including environmental conditions, predator pressure, and the availability of resources. Parental care is essential for passerine reproductive success. Passerine chicks are altricial, meaning they are born naked, blind, and helpless, requiring constant attention and feeding from their parents. This extended period of parental care has likely contributed to the evolution of complex social behaviors in some passerine lineages, such as cooperative breeding, where multiple individuals help raise a single brood. The evolutionary history of passerines, illuminated by a combination of fossil evidence and genomic analysis, tells a story of ancient origins and remarkable diversification. The current understanding suggests that passerines first emerged around 47 million years ago in the South Pacific, near what is now Australia and New Zealand. From this ancestral homeland, they radiated outwards, colonizing new continents and diversifying into the three main suborders we recognize today. Acontheceti, New Zealand wrens, Turani, Sabosons, and Passerine, os signs or songbirds. The spread of passerines across the globe has been influenced by geological events, including the separation of continents and the formation of new land bridges. The emergence of Wallachia, a group of islands between Australia and Asia, played a crucial role in facilitating the dispersal of passerines from their Australian homeland into Asia and beyond. This geological event, driven by tectonic plate movements, created a stepping stone pathway across what had previously been a vast expanse of open ocean. The fossil record, though fragmentary, provides tantalizing glimpses into the biogeographic history of passerines. The discovery of Phoropinarium, a stem turrico in the Eocene of North America, challenges the traditional view of a strictly Gondwanan origin for this group, highlighting the complex interplay of dispersal, extinction, and vicariance in shaping their distribution. While modern turricos are restricted to Africa, the presence of their Eocene relatives in North America suggests a much wider distribution for this lineage in the past. Genomic analysis, using techniques like the comparison of ultra-conserved elements and mitochondrial DNA, has revolutionized our understanding of passerine relationships. This powerful tool has helped resolve long-standing debates about the passerine family tree, revealing surprising connections and overturning traditional classifications. The ongoing research, through projects like Open Wains, continues to refine our understanding of passerine evolution, promising even more detailed insights into the intricate relationships within this extraordinary group. The story of passerines is a symphony of feathers, a testament to the power of evolution. Their small size, their mastery of perching, their diverse nesting strategies, their complex vocalizations, and their ability to exploit a vast array of food sources have all contributed to their remarkable success. From their ancient origins in Gondwana to their current global dominance, passerines have woven a rich and vibrant tapestry of life, inspiring awe and wonder in all who encounter their diverse forms and behaviors. Chapter 7. Open Wings, Open Questions, the Future of Passerine Research. Passerines, the perching birds, represent an evolutionary triumph. Their diversity, encompassing over half of the world's bird species, their songs that fill our skies, and their presence in nearly every corner of the globe, all speak to a remarkable evolutionary journey. Yet, despite the impressive strides made in understanding their history, many intriguing questions remain unanswered, beckoning researchers to delve deeper into the passerine enigma. The perching foot, a defining characteristic of this group, is a marvel of biomechanical engineering. 3 toes pointing forward and one robust, opposable hind toe, or hallux, allow passerines to securely grasp branches of varying sizes. But how did this unique foot structure evolve? Current theories suggest a gradual transformation from the feet of their terrestrial theropod dinosaur ancestors. Fossil evidence from early Eocene birds like Eozygodactylus americanus reveals an intermediate stage in this evolution, with feet showing an increasing ability to grasp, a crucial step towards the refined perching abilities of modern passerines. Biomechanical studies, using techniques like high-speed video and force plate analysis, are helping to unravel the intricate workings of the passerine foot, revealing how the muscles, tendons, and bones work in concert to allow for effortless perching, even during sleep. Further research exploring the evolutionary pressures that may have driven the development of this specialized foot, such as the need to access new food sources in trees or escape from ground-dwelling predators, promises to shed more light on this key adaptation. The extraordinary diversification of passerines, their radiation into over 6,000 species occupying a vast array of ecological niches, is another puzzle that continues to fascinate researchers. While factors like diet specialization, competition, and changing environments undoubtedly played a role, the specific ecological triggers and evolutionary mechanisms driving this explosive diversification remain elusive. Some passerine families, like the ovenbirds, Furnariidae, of South America, have diversified in extraordinary ways, exhibiting a remarkable range of nest types and foraging strategies. From the mud oven nests of the Rufous hornero to the simpler domed nests of foliage-gleaning Furnariids, each species has adapted its nest and foraging behavior to its specific environment. Scientists are using ecological data and sophisticated modeling techniques to understand these diversification patterns, exploring how factors like resource availability, predator-prey interactions, and climate change have shaped passerine evolution. Migration, a defining characteristic of many bird species, also presents intriguing questions for passerine researchers. Lesser kestrels, Falco naumani, for instance, exhibit a fascinating pre-migratory behavior, gathering in large roosts in the Balkans before their long journey to Africa. Genetic studies using microsatellite markers, analyzing feathers shed at these roosts, have revealed that birds from multiple breeding populations converge on these sites, some traveling hundreds of kilometers. This research not only highlights the importance of pre-migratory sites for these birds, where they can replenish energy reserves and undergo molt, but also raises broader questions about the evolutionary origins and genetic basis of migration in passerines. How do these birds navigate across vast distances? What are the genetic and environmental cues that trigger their migratory journeys? Further research into the genetic and physiological mechanisms underlying migration promises to unravel the mysteries of this remarkable avian behavior. The vocal prowess of passerines, their ability to produce complex songs and mimic a wide array of sounds, is another area of active research. Aucens, the songbirds, possess a unique vocal organ called the syrinx, located at the base of the trachea. This complex structure, with its independently controlled membranes and muscles, allows songbirds to produce two distinct sounds simultaneously or rapidly switch between notes. Genetic studies have revealed that the ability to detect sweet tastes, important for nectar-feeding birds, arose early in songbird evolution, possibly in Australia. This discovery, alongside other evidence for an Australian origin of songbirds, raises questions about the ancestral environment and selective pressures that shaped the evolution of both vocal learning and taste perception. Studies exploring the structure and function of the syrinx, the neural pathways involved in vocalization, and the genetic basis of vocal learning, are shedding light on the evolution of this remarkable communication system. The extraordinary mimicry of species like the superb lyrebird, capable of imitating an astounding range of sounds, further underscores the complexity and adaptability of the passerine vocal apparatus. The timing of passerine origins and their early diversification has been a subject of intense debate. While some researchers have proposed an ancient Mesozoic origin, coinciding with the breakup of Gondwana, the scarcity of Cretaceous passerine fossils poses a challenge to this view. The KPG extinction event, a catastrophic event that wiped out the non-avian dinosaurs 66 million years ago, may have played a significant role in shaping early passerine evolution. Did this extinction event create new ecological opportunities for passerines, allowing them to diversify and radiate into newly available niches? Or did it decimate early passerine lineages, leading to a more recent Cenozoic origin for the group? The current consensus, based on genomic data and fossil calibration, places the origin of passerines in the early Eocene, around 47 million years ago. This more recent timeline suggests that the KPG extinction may have had a less direct impact on passerine origins, although it could have still influenced their subsequent diversification. Further research, integrating fossil evidence, genomic analysis, and ecological data, is needed to fully understand the interplay between mass extinction events and the evolutionary history of passerines. Beyond these core questions, numerous other mysteries remain. The evolution of specialized behaviors, like brood parasitism in some passerine families, such as cuckoos and cowbirds, where one species lays its eggs in the nest of another, raises questions about the evolutionary arms race between parasites and their hosts. How do brood parasites mimic the eggs and calls of their hosts? How do hosts evolve defenses against parasitism? Another area of interest is the role of passerines in ecosystem processes, such as seed dispersal and pollination. Many passerines, like the green-headed tanager and the tui, feed on fruits and nectar, playing a crucial role in the reproductive cycles of plants. How have these interactions shaped the evolution of both plants and birds? What are the ecological consequences of disruptions to these mutualistic relationships? The future of passerine research is bright, with new technologies and collaborative projects promising to further illuminate the evolutionary history of this remarkable group. Advanced genomic techniques, sophisticated fossil analysis, detailed behavioral studies, and innovative research projects like Open Wains are providing unprecedented insights into the passerine family tree. As new discoveries emerge, they will undoubtedly rewrite chapters of their evolutionary story, deepening our appreciation for the intricate web of life on Earth and the remarkable adaptive radiation of these feathered wonders. Passerines, or perching birds, represent an astonishing evolutionary success story. From their ancient origins on the supercontinent Gondwana to their current global dominance, these feathered wonders have diversified into over 6,000 species, occupying nearly every terrestrial habitat on Earth. Their small size, specialized perching feet, diverse nesting strategies, complex vocalizations, and adaptable diets have all contributed to their remarkable radiation. Genetic analysis has revolutionized our understanding of passerine evolution, revealing surprising relationships and overturning long-held assumptions. The discovery of a new family of passerines in Africa, the revised timeline for their diversification, and the confirmation of an Australian origin for many key traits, all highlight the power of genomic tools in rewriting evolutionary narratives. Fossil discoveries, though rare and often fragmentary, provide crucial calibration points for molecular clocks and offer glimpses into the morphology and distribution of extinct passerine lineages. The discovery of a stem turrico in North America, for instance, challenges traditional biogeographic narratives and suggests a more complex history of dispersal and extinction. The remarkable adaptability of passerines is evident in their nesting behaviors, vocalizations, and even their taste receptors. From simple cavities to elaborate woven structures, their diverse nests reflect their ability to exploit a wide range of habitats. The evolution of the syrinx, a unique vocal organ, has enabled the complex songs and mimicry that characterize many passerine species. Even their sense of taste has adapted to their environment, with some lineages regaining the ability to detect sweetness, crucial for exploiting nectar and fruit resources. The ongoing research on passerines promises to further unravel the mysteries of their evolutionary journey. Studies exploring the biomechanics of the perching foot, the ecological drivers of their diversification, the genetic basis of migration, the neural pathways involved in vocal learning, and the interplay between mass extinction events and evolutionary history, are all contributing to a more complete picture of this remarkable group. The story of passerines underscores the interconnectedness of life on Earth and the power of evolution to shape diversity. From their ancient origins to their current global dominance, these birds have adapted, diversified, and thrived, leaving an indelible mark on our planet's ecosystems. Their story is far from over, and ongoing research promises to reveal even more fascinating details about their evolutionary journey. What questions do you have about passerine evolution? Share your thoughts and curiosity in the comments below. We encourage you to like and subscribe to stay updated on our latest explorations into the natural world. And if you have suggestions for future topics, we'd love to hear them. Remember, the journey of discovery is a continuous one, fueled by our innate curiosity and desire to learn.